Hello students, in this class we are going to discuss the structure of DNA. The structure of DNA. Normally, the structure of DNA is made up of, basically it is made up of three basic components. Out of these, the first one is nitrogen bases, the second one is ribose sugar, and third one is phosphate group. One by one we are going to discuss the structure of DNA. Basically the DNA is made up of whatever the structure of DNA. Over the structure of DNA, the genetical information is present in the form of nitrogen bases, and there are four type of nitrogen bases are present. One by one we are going to study the basic nitrogen bases are present. So, which are the basic different type of nitrogen bases are present? Out of these, the two are purines, while two are pyrimidines. So look at this, it is very interesting. So, which are the purines? Adenine and guanine. Cytosine and thymine. So what are the purines? Purines are adenine and guanine. What are the pyrimidines? Cytosine and thymine. Okay. So as we know that inside the book there is a language. Inside the book there is an English language, and the English language is made up of 26 alphabet letters. That is A, B, C, D. Okay. In this way, the information which is stored over the structure of DNA, that information is stored in the form of nitrogen bases. Okay. So here the information is stored in the form of purine and pyrimidines. Here look at this. Here there is a two strands of DNA. Two different strands of the DNA. So look at this, how it is complexly arranged with each other. As we know that both the strands are anti-parallel to each other. Look at this, anti-parallel means what? One strand, both the strands are running opposite to each other. One strand is running 5' to 3' direction, while another strand run from 3' to 5' directions. Means both the strands are moving opposite directions. So when we discuss purines and pyrimidines, okay, this adenine, guanine, cytosine and thymine. Okay, so you just have to keep one thing in your mind, that adenine always pair with thymine. While cytosine always pair with guanine. And in between them, purine and pyrimidine, okay, adenine always pair with thymine, and there is a two hydrogen bonds are present in between them. Okay, in this way, the guanine or cytosine always pair with guanine, or guanine always pair with cytosine, and there is a three bonds of the hydrogen. Three hydrogen bonds are present in between cytosine and thymine. Okay, so what we discussed, that the structure of DNA is basically made up of three basic components. Out of these, one is the nitrogen basis, while the second one is sugar. So you all of you know that what kind of sugar or which sugar is present inside the structure of DNA. When we see the long form of the DNA, that is deoxyribose nucleic acid. So it is clear that inside the structure of DNA, ribose sugar is present, and we are going to learn the second component of the structure of DNA, that is ribose sugar. So listen, what is ribose sugar is going to tell us, that is, okay, this is the ribose sugar. Okay, it contains 5 carbon atoms. 1, 2, 3, 4 and 5. So therefore, this ribose sugar is also known as pentose sugar. Okay, why? Because it is having 5 carbon atoms. So therefore, this ribose sugar is also known as pentose sugar. Pentose sugar. Okay. So again, the third one component is phosphate group. Phosphate group. Okay. So one thing that you have to keep in your mind, that this phosphate group, okay, the source of this phosphate group is always H3PO4, means phosphoric acid. Phosphoric acid. The source of the phosphate group is always phosphoric acid. And how? Whenever there is a formation of the structure of the DNA, these 3 basic components are used. These 3 basic components are used. Okay, but these are the separate form. These are in separate form. When there is a formation of DNA, structure of the DNA, these 3 components are assembled with each other. Assembled with each other. So one thing that you have to keep in your mind, that the structure of DNA inside the eukaryote, it is made up of polynucleotide chain. Polynucleotide chain. Okay. So how it is made up? It is very interesting. Look at this. As we earlier discussed, that the structure of DNA, it is made up of nitrogen bases, and it is having which sugar? Ribose sugar, and it also having phosphate group. So what we studied, that each strand of DNA is made up of nucleotide. Nucleotide. So let us see the structure of nucleotide. The structure of nucleotide. What we see? The structure of DNA. It is made up of basically 3 components. Let us take sugar first of all. Which sugar? Ribose sugar. And ribose sugar is a pentose sugar. See, this is the pentose sugar. Okay. Now, this is the nitrogen base. This is the nitrogen base. And over the 5th carbon atom, there is an attachment of phosphate group. There is an attachment of phosphate group. See, when inside the nucleotide, phosphate group is present, sugar is present, and nitrogen base is present. 3 components are present, we called nucleotide. But if we remove the phosphate group, we called it is nucleoside. Nucleoside. Okay. So you all of you got the difference in between nucleotide and nucleoside. When we see there is an attachment of the phosphate group, we called nucleotide. But when we remove the phosphate group, it is nucleoside. But when there is a construction of the DNA strand, nucleoside is not used, but nucleotides are used. What we say, the statement is very important. The statement is very important. Listen what I said, that each strand of DNA is made up of polynucleotide chain. The name itself indicates poly means more than one. See, this is the phosphate group and this is the nucleotide. Let us see how there is a construction, exactly the construction of the DNA strand. Look at this. It is very easy. See, this is one nucleotide, this is another nucleotide. Okay, and there is a formation of a special type of chain, of the structure of DNA. See, okay, here the nitrogen base is present. Nitrogen base is present. As we already discussed, what is meant by nitrogen bases and which are the nitrogen bases? Adenine, guanine, cytosine and thymine. Adenine, guanine, cytosine and thymine. You all of you know that adenine always pair with thymine. Adenine always pair with thymine with 2 hydrogen bonds. Guanine always pair with cytosine with 3 hydrogen bonds. Cytosine always pair with guanine with 3 hydrogen bonds. Thymine always pair with adenine with 2 hydrogen bonds. Okay, so this is the one strand, but we see inside the eukaryotes there is a 2 strands, and both the strands are present in opposite direction. Both the strands are run opposite direction, means they are opposite as well as anti-parallel to each other. Anti-parallel to each other. See what exactly happened here. As we see the structure of eukaryotic DNA. The structure of eukaryotic DNA. Okay. So there are two different type of strands, and as we see the construction of DNA strand, because as we know that a statement, that is each strand of DNA is made up of polynucleotide chain. Polynucleotide chain. Here there is a formation of chain because one nucleotide is attaches with other and one nucleotide is attaches with other. So therefore there is a formation of chain of nucleotide. Chain of nucleotide. So listen guys, this strand is run from 3' to 5' direction. So obviously another strand will run from 5' to 3' direction. 5' to 3' directions. Okay. So both the strands are running opposite direction and both the strands are anti-parallel to each other. Anti-parallel to each other. Okay. So this one is the part of structure of DNA. Okay. So next part, let me discuss some interesting phenomena and interesting facts regarding the DNA. Okay, all of you know that the DNA is having ability to store the entire information regarding our body structure. So therefore, all of you know that a small cell and its nucleus is having information regarding the body structure. Okay, so all of you know that what is meant by heredity. Heredity is nothing but transmission of genetical information from one generation to another generation. One generation to another generation. Why heredity is happened? Why inheritance is happen? It happens because the DNA is having ability to store the information and it carries from one generation to another generation in the form of chromosome. In the form of chromosomes. Okay, what is that? What is the role of these chromosomes? Chromosomes are nothing but condensed form of DNA. Condensed form of DNA, and they are nothing but they are carrying information from one generation to another generation. Okay, so guys, in this lecture we are discuss the structure of DNA, nucleotide, nucleoside, the basic components of the structure of DNA and how the DNA is constructed. Okay, and we also discuss some interesting facts regarding the structure of DNA. So I hope you enjoyed this lecture.